Hi everyone, my name is Tori Bauer-Pisani. Today, July 23rd, 2023, I am presenting two case studies that pertain to the material we have learned so far in this course. The first case study concerns a 16-year-old female that is recently diagnosed with a deficiency of muscle glycogen phosphorylase. Both her and her family are concerned and have several questions pertaining to the new diagnosis. They have asked four primary questions in which I will answer by outlining the altered processes of skeletal muscle glycogenolysis and muscle metabolism. The first question the patient asked was whether she can take long walks with her friends, which she really likes doing. My answer to that question is that she will experience exercise intolerance and extreme muscle cramps. The disease typically associated with muscle glycogen phosphorylase deficiency is also known as McArdle disease, which is an autosomal recessive metabolic disorder. Glycogen phosphorylase is the key enzyme used in the first step of glycogenolysis to release glucose 1-phosphate monomers from the intracellular glycogen stores. Glycogen is essential to provide energy to skeletal muscle tissues. Because the glycogen phosphorylase enzyme isn't present to catalyze the reaction that breaks down glycogen to use in the muscle tissues, fatigue, muscle cramps, and pain is experienced after short duration of exercise. There is a chance that the patient will develop something called second wind after taking a brief break after starting exercise. This means that they will be able to resume exercise after that break without any discomfort. Based on this information, I would say that this patient should be able to take walks with her friends, but should be aware that her tolerance may not be able to withstand long, strenuous durations. The second question pertains to whether the patient should be worried about having reduced muscle glycogen stores. Based on our knowledge of the disease, there is a deficiency in the enzyme that begins the breakdown of glycogen and not the reduction in the amount of glycogen stores. If anything, there is an overabundance of glycogen within the muscles that is trapped. This accumulation can eventually lead to rhabdomyolysis, or the death of muscle cells that consequently releases toxic contents into the bloodstream. The third question the patient asked was whether she needed to get up in the middle of the night to eat to maintain her blood glucose levels. The short answer is no. During an overnight fast, the liver synthesizes glucose from compounds, which does not include the glycogen phosphorylating enzyme involved in glycolysis for muscle cell energy. However, there is some evidence that consuming a carbohydrate-heavy meal before exercise helps to improve muscle performance in those with McArdle disease by improving the supply of glucose to the muscle tissues. The glycolysis of glucose can be broken down by muscle tissues still. Maintaining glucose should not be an issue for those with muscle glycogen phosphorylase deficiency. However, some instances of hypoglycemia have been noted. Finally, the patient asked whether she should be concerned with production of excessive lactate during intense anaerobic exercise. The answer is no. There is a lack of lactate produced because glycogen is unable to be broken down in the muscle cells to catabolize to lactate. And this concludes the first case study this week. I will now move on to the second case study, which we aim to understand how the regulation of glucose is utilized by different tissue cells. The case presented shows images generated from an MRI and PET scan of a brain with a large necrotizing glioblastoma. The question is, how does glucose uptake in normal tissue compare to glucose uptake by tumor cells in the above images, and why would this be advantageous to the invasive tumor? Firstly, we need to understand how glucose is utilized in normal tissues. We have several metabolic pathways in our bodies in order to utilize glucose, which include glycolysis, glycogenolysis, pentose phosphate pathway, and the citric acid cycle. These are used in either fed or fasted states to fuel our tissues. Much of our glucose comes exogenously via food. Carbohydrates are digested in the small intestine, and those polysaccharides are absorbed into the bloodstream. The concentration of blood sugars are met by 3 regulatory hormones: insulin, glucagon, and epinephrine. If blood glucose is increased, then insulin from the pancreas is released to transfer glucose into liver and muscle cells, where glucose is converted to glycogen for energy. In normal glucose catabolism, glucose is reduced to pyruvic acid, where the energy released is converted to ATP and further reduced to NADH. This is called glycolysis, in which glucose is catabolized either with or without the presence of oxygen. With oxygen, energy is converted to acetyl-CoA to be utilized by the citric acid cycle to generate more ATP, NADH, and FADH for cellular energy storage. Without oxygen, glucose is converted to pyruvate, which is further converted to lactate. The difference in cancer cells is that they are extremely demanding in their need for energy. Therefore, their requirements lead to inefficiency in the way glucose is utilized. Cancer cells rely on glucose to fuel aerobic glycolysis to generate ATP. The earliest report for the role of glucose metabolism in cancer cells was made by Otto Warburg in the early 1900s. He stated that cancer cells utilize aerobic glycolysis even in the presence of oxygen to form lactate, which, as we know from normal respiration, is incorrect. This is highly inefficient in the presence of normal functioning mitochondria. The phenomenon can be explained by the fact that cancer cell proliferation causes alterations in cell signaling pathways that are crucial to the maintenance of metabolic pathways. With this metabolic change, substrates for cancer cell proliferation and division are now provided by having high carbon sources for anabolism, rapid ATP availability, an abundance of lactic acid to increase redox reactions via the glycine-serine pathway. Therefore, the tumor is continuously wanting to proliferate. Thus, the demand for energy via glucose is very high. More glucose means more fuel to expand tumor cells. This phenomenon explains why we can visualize a tumor in the PET scan image after administration of fluorescent D glucose. Cancer cells are catabolizing glucose aerobically and in higher volumes than other normal tissues in